Good afternoon, everybody, and welcome to our webinar on sequencing the coronavirus genome. Fantastic to have so many of you here. You've all successfully navigated the technology, which is great. I guess we are all getting fairly used to meeting on Zoom these days, so this is fairly familiar territory. So we're going to have a quick look at our coronavirus sequencing solutions here at YouSeq. My name is Jim Wicks. I'm the CEO of YouSeq, and I'm joined today by Tom Huckvale, who is our senior scientist who has led the project to develop this new product in double quick time during the coronavirus outbreak. YouSeq is a business that has been set up specifically to make next generation sequencing easy to understand and easy to execute. And there has never been a time when we have needed this technology more. It's going to be critical to sequence the genome of this virus as it spreads around the world. And we therefore believe that our mission to make this as easy and as accessible as possible has never been more pertinent than it is right now. So why do we need to sequence this virus? Well, we are already seeing very, very large numbers of sequencing files appearing in public databases, and that is indicative of how important this screening and monitoring project that we're seeing across the globe really is. I predict this will become the most sequenced piece of certainly pathogen DNA in history very, very quickly. The reason we need to monitor any changes in the genome of this virus are fairly obvious. Mutations can affect the biology of the virus. Now, it's an RNA virus. RNA viruses do mutate, is what they do. It's a key part of their evolutionary success story. So we're already seeing mutations occurring in the lineage as the virus transmits around the world, and we should expect to see more, but we need to be keeping a really close eye on it in case anything very dangerous occurs. And we're most worried about mutations affecting, literally how dangerous the virus is, affecting the health outcomes for anyone infected. So that needs to be monitored. Of course, people are scrabbling to develop treatments and therapy for treating the virus. We're just starting to see first regulatory approvals for certain drugs that are being repurposed for treating the virus. And there'll be others in the pipeline. But of course, if the virus mutates, which RNA viruses do, it's likely they will mutate and evolve away from those therapies, and those therapies will become less successful. So the key to keeping good quality therapies in our hospitals is to understand what's happening with the virus itself. And of course, we're all very interested in how quickly this virus is spreading around the world. The UK government is constantly measuring what they're calling the R value, which is essentially a measure of how infective the virus is, how many people the virus is being passed on to when someone is infected. That number could change dramatically if the virus was to become more infective, if mutations were to occur that had an impact on how successfully it can spread. That would be very, very serious indeed. And of course, the globe is on tenterhooks until we have a vaccine for this virus. And those vaccines all rely upon antibodies recognizing the surface proteins of this virus. And of course, if there were mutations that occurred that led to changes in those surface proteins, it could seriously affect the efficacy of any of those candidate viruses. So it's utterly critical that around the globe, as a global community, we are monitoring the genome of this virus. And that's exactly what we are setting out to help with here at YouSeq. We've built a kit that reveals really powerful information about the virus genome. It's easy to use. If you've ever done any molecular biology before, you'll be able to use this test, even if you've not done sequencing before. Critically, with all our products, we always provide everything you need in one kit. So it's just one catalogue item to order. Lots of our competitors, you have to buy primers from one place or enzymes from another place, or a different catalogue number for your cleaning reagents and a different catalogue for reverse transcription. We just feel like that's far too complicated, and what you really want is a kit that has been built and optimized to work in one neat workflow. So that's what we do. Everything you need to make sequence-ready libraries in a very quick and simple protocol is provided in the kit that we're producing. And I'm going to talk you through each of those components in a bit more detail. But alongside that, the most daunting part of next-generation sequencing is not actually the bench work. It's actually the data analysis. It can be complicated. So we felt it critical in this crisis situation that it's as easy as possible to process and understand the data, to find out what's going on, with the sequence of the virus, without needing a PhD in bioinformatics to do so. So we have built a free of charge, free to use, online bioinformatics tool, and it is as simple as loading your FASTQ files, so that is the sequencing files you get from your Illumina sequencer, into our cloud. Click the submit button, and in around 2 minutes you'll see results. We are going to show you that in more detail during this webinar as well. But it means that we've built an end-to-end solution. So we have a library preparation kit. It's currently viable for use on Illumina instruments, but we hope to launch an Ion Torrent version soon too. And then we have a very fast, ultra easy to use analysis tool for looking at the data afterwards. So we're going to walk you through a real world example of this test in use. So this is a real test that we have done here in our laboratory. We've done lots of validation, of course, but this is just one example from one particular swab that we were given. And of course, this is what really matters. I could show you lots of predicted performance data for our kit about coverage and depth, etc., etc. But we tend to try and avoid using too much jargon when we're talking about next generation sequencing. It's a very jargon-filled subject and can often therefore be very difficult to penetrate to, very difficult to understand, particularly for new users. So we try to keep our language really simple. And of course, what we really care about in this situation is not any of that jargon, not any of the complications. We care about is, can we take a clinical swab from someone who's tested positive for coronavirus, and can we produce meaningful, useful, powerful next generation sequencing data? So that's the story that we're going to tell you now, a real clinical swab and how you use our kit to get sequencing data from it. So the swab that we're going to talk about is one that we've taken from Southampton General Hospital. They've been really supportive in helping us to develop this test. They understand how important this kind of science is going to be. And they've given us several positive samples from the hospital. But on this occasion, we've deliberately chosen one with quite a low viral load, because this is what the vast majority of swabs in the world that you're likely to come across will contain. We have seen swabs with terrifying viral loads on them. We've seen CT values by qPCR of less than 14, representing hundreds of millions of copies of the virus per sample. But they're rare, thankfully. The vast majority look a bit like this. You tend to see quite late signals. So this is a qPCR test for this particular swab. It's a three-way multiplex detecting the E gene, the RDRP gene, and the N gene of the virus, alongside a human endogenous control. So this is a qPCR test that we also produce and supplying in large numbers at the moment too. But it gives a CT value, as you can see, of around about 31, 32, the way those amplification plots are taking off. And that equates to around 800 copies of the virus in this sample. So it's quite a low viral load. And if we can get good quality sequencing data from this kind of sample, then you can be really confident you can get good quality sequencing data from just about any sample. So the workflow, what do you actually do at the bench side? What does it look like? Well, it takes around about 3 hours of hands-on time to produce a sequencing library. We are routinely producing libraries comfortably inside a working day here at YouSeq every day at the moment. Very, very simple protocol, and I'm going to walk you through that. So the first step on the journey is to take the RNA from the swab. So any extraction method that you're familiar with will be absolutely fine. We're big fans of magnetic bead-based methods, but QIAGEN spin columns, etc. All the things that you're familiar with will be fine to extract RNA. And then we have a very simple reverse transcription step to turn that RNA into cDNA. So if you've ever done a reverse transcription before, you'll be very familiar with this kind of protocol. We use random primers to prime the RNA, and then a reverse transcriptase, a very powerful one that operates at quite a high temperature to untangle the viral RNA, to ensure you get a really efficient one-to-one conversion of your RNA into cDNA. That's about 5 minutes of hands-on time, very, very easy to do. After that, it's about setting up a PCR reaction, and I'm sure all of you will have set up a PCR reaction along the way. You set up 2 reactions per sample, so we have a pool A and a pool B to make sure we don't have overlapping amplicons. A very, very simple reaction. There's master mix, there's your sample, and then there's the addition of our primers, which have been carefully designed to deliver very short amplicons. So we're aware this is an RNA virus. RNA is very prone to degradation. So for that reason, it's better to have as shorter amplicons as possible. So essentially you set up 2 PCR reactions for each sample. This can be done on a 96-well plate on any PCR machine that you have available. And then it's very quick. It's only 24 cycles of PCR where the entire genome will be essentially fragmented and amplified slightly to increase the sensitivity of your test. After that, there's a cleaning step. So you want to remove any unwanted artifacts and any leftover biological material in your sample at this stage. So we have a proprietary enzyme blend that we use to treat the product at this stage to remove artifacts, and then a very simple magnetic bead cleaning step. And again, all of these reagents are provided in the test. I'm sure most of you will have worked with magnetic bead cleanup steps before. Very, very simple to do. This has also been built to be fully automatable, so this is robot friendly. All the volumes involved in all of this protocol are large enough for robots to pipette very, very accurately indeed. In fact, we're using robotics here in the laboratory every day to run this protocol. So, simple cleanup step, then you go into another round of PCR, and this is the round where the adapters and indexes are attached. So again, the kit is supplied with indexes and adapters. You don't need to buy those separately. You don't have to go and go get those elsewhere. It's all in the kit. So we have a very quick 8 cycles of PCR where your indexes and adapters will be attached to the amplicons that you've made in PCR1. Simple as that. And then there's a final cleanup step. No enzymes this time for this cleanup step because you've really got quite a pure library at this stage. It's just about removing any last little bits and pieces you don't want going into your sequencing reaction. It's just a simple magnetic bead size selection exercise. And then your library's ready. So the final step is just to quantify that. So you'll have multiple samples on the go here, I'm sure. And you want to make sure you put the same amount of each of those, the libraries from each of those samples into your pool to go onto your sequencer. So we recommend quantifying your libraries at this stage. You can do that very simply with a qubit or similar sort of fluorimeter-based method. Or we also sell a qPCR test for quantifying next generation sequencing libraries. Very quick, very easy to do, very, very accurate, the qPCR method. But essentially you then want to make sure you're putting the same amount of each of those libraries into a pool and it is ready to load onto your sequencer. This is what your library will look like if you have a look at it by bioanalyzer. That's what we've done here. So we have a tape station and we've been looking at these libraries that way. You can see a really, really nice clear band of around about 350 bases. These amplicon-based libraries tend to produce these nice narrow libraries. All of the inserts are of a very similar size, so you see these nice neat libraries of precisely the right size. You can see lovely and clean, no primer dimers, no artifacts of any significance larger than the library itself. And this is what you'd expect to see with our test. And this library here has been produced, as we said, off only 800 copies of the virus. Really, really beautiful libraries, ready to be sequenced. And at the moment, we have built this with adapters and indexes for the Illumina platforms, just because that is what the vast majority of people in the world are using right now. We could very easily develop an ion torrent version of this protocol if you're interested, or potentially other platforms as well. So it's Illumina right now, but contact us if you're interested in using this method on any other platform. But you simply load your library onto your instrument and set that off, and then you will receive some data. Now, of course, data can be the part that is perhaps the most daunting of all, but we wanted to really simplify that process. So we have built this end-to-end solution. And I'm going to hand over to my colleague Tom. So Tom Huckvale is our senior scientist running this project here at YouSeq, and he is going to walk you through our free of charge, easy to use data analysis pipeline.
Thanks, Jim. So I'm going to have a quick run through of our data analysis pipeline. So this was the page that you will start with. So you've just used our kit and generated some libraries, and these libraries have been sequenced on an Illumina platform. So what you'll do is you take the demultiplexed file, the FastQ file from your machine, and you'll just plug it into this data analysis tool. So if I just choose reads that we did recently, if I choose some samples. So here goes read 1 and then the read 2, FastQ. And so these are from clinical samples that we've got from a local hospital. So I'm just going to submit this job to the data analysis pipeline, and it'll come up with a progress bar here. So it will submit the job and upload these FastQs. So these FASTQs are actually quite large. I think this is about 2.5 million reads. And the FASTQs here are about 90 megabytes. So the first thing it'll do is it'll align and sort these sequences. And this sort of takes a moment. And it'll be aligning these sequences to the coronavirus genome. So just give this a minute to work through. So these samples came from a local hospital, and they weren't hugely concentrated in terms of the viral load. They were pretty low, had a pretty low CT value. So they're actually quite weak samples, but with this kit, because you're using this amplicon-based approach, you can really generate libraries from very, very small amounts of starting material. And so now the software is trimming the primers, and it's important to make sure that in your final sequences, your final consensus sequence, doesn't include any primer sequences, especially if you're looking for variants. And so this is a fairly key step in our analysis pipeline. So in your finished data, you can be sure that the sequence you have comes directly from the template and not from our primer pools. And again, this will take a moment to run through. And next it'll sort the trimmed primers, and it'll index them as well. And then it's going to calculate some depth and coverage metrics. And this is generally quite a fast process in the pipeline. And really this is now going to generate you some nice plots and some tables of just how much of the genome, the viral genome, has been covered in this sequencing run. And then the job has finished. So if we just close this, and as you can see, we can achieve with this kit very uniform coverage of the entire viral genome. So at about 10 times coverage, we have the entire genome covered using this primer set in our NGS kit. And what it'll also give you is it'll give you a nice package to download as well in a zip file. So if we just download this now. And so here we have the contents. So the data analysis gives you some high resolution images, so plots you can use, as well as some tables. But what it'll also give you is it'll give you some coverage based on each amplicon as well in a CSV file, and also some text files as well to use for further data analysis. But what it'll also give you is indexed BAM files, which you can then use to view your reads aligned to the genome in a genome viewer of your choice. So I'm just going to quickly demonstrate one of these clinical samples in IGV. So if I load IGV and make sure that I have the coronavirus genome loaded, and IGV is just a free tool, freely available to download. So if I then load the file that we've just grab the file from our data analysis pipeline. So if I take the primer trimmed and sorted file, and this is quite, I think this sample had over 2 million reads, so it's going to take a moment to load into IGV. This is certainly on the larger side of the read number. I think you normally expect something a bit lower. I think it's not necessary to sequence at this particular depth. So we'll just wait for this to load. I think it's maybe around 2.5 million reads for this sample. Okay, so there you have it. So here you can see all of the reads now aligned to the coronavirus genome. And what we'll be able to do is, using this tool, we'll be able to browse it. So here you can see the full 30 kilobase genome and all of the reads, the sequencing reads from our run. So if we can zoom in to see if we can find some interesting variants from this particular clinical sample, and in particular we're going to look, I think we've found a few in the spike protein region here, which is certainly of interest, especially in terms of controlling the virus. And as you can see, there are differences to the consensus. So in particular, There's a variant here at around 23.4 kilobases, and you can see a definite variant from the consensus. So the consensus will see an A, and here in this particular clinical sample we're seeing a G. And certainly this is pretty routine from what we've seen from clinical samples. Certainly in your geographic area there are going to be differences, and this is certainly a good example of just the power of NGS to drill down into some of these particular variants. So here's another one here. You can certainly see in all the reads, in the forward and reverse reads. So you have quite high confidence, very high coverage. So you certainly have very confident in a lot of variant calls that you have. Okay, and back to.
Thank you very much, Tom. Really nicely explained. I hope you can see just how easy that pipeline is to use. And even if you've never done anything bioinformatics-wise before, we really believe we simplified that to a stage where even beginners could tackle this sort of thing themselves. Really powerful, really, really easy to use. So that concludes our webinar, really. A little bit of information here just on the screen, just showing the performance characteristics of the test. We are seeing greater than 99% genome coverage, which is fantastic, very uniform, so we're not seeing PCR bias of any meaningful nature in this test at all. And it makes for a very, very sensitive test, and that's what really counts in this situation. We want to be able to get libraries from as many swabs as possible, as many different types of clinical samples as possible. Very nice and easy, of course, if you've got a very high viral load, but even with low viral load samples, you should be able to expect good quality sequencing data. And over here on the left, you can see a prediction of the approximate number of samples you could get on a run with all the typical Illumina instrumentations. And I think, of particular interest, the MySeq is the best-selling instrument out there, and lots of you will have MySeqs. And the MySeqs with the nano or the micro chemistry on board make for a very affordable method for the actual sequencing step as well. And of course, there is potential for very, very high throughput sequencing as well. And if you are into that world and you would require a kit from us with a larger number of indexes and adapters, that's something we could do. So do please contact us about that if that is of interest. So that is our solution. That's our contribution to the sequencing challenge that the globe is facing right now. It's very fast. It's very easy to use. We're really excited to see it being used around the world to contribute to this situation. It's probably worth mentioning just here that we do also have a fantastic range of multiplex qPCR tests for coronavirus as well. We believe that those multiplexing tests offer real additional levels of security, detecting 2 or 3 targets at once to be certain of your diagnostic result, to be really powerful. So if you're interested in qPCR tests, that's something we can talk to you about as well. But we're here to talk about next generation sequencing right now. If you have any questions, we're going to jump into a live Q&A session now. Or if you want any more information or would like to ask your question later on today, please feel free to drop us an email asking any of your questions to hello@youseq.com, and we will pick those up and get back to you as quickly as possible. But let's jump over to a live Q&A now. So you can use the chat feature there. You can submit your questions, type your questions in, and we will answer as many of those as possible over the next 5 or 10 minutes.